Change gears a little bit. We've been talking about models of evolution. And remember at the start I emphasized special case models, there's a certain sort of format to it. You have some complicated system you want to study. And the mindset of model building is always, it's so complicated. We're never going to be able to come up with a mathematical description of it just because it's so complicated. So we'll simplify it, right? We'll make simplifying assumptions, and then it'll be easier to work with. And so in model building, you start out with simplifying assumptions, and then you start doing math and building a model out of it. You don't have to do that. We're going to look at the other approach today and then be applying this through the next couple of lectures. So first of all, we're going to use slightly different math. And this is stuff that I think everybody's familiar with, but it's so core to evolutionary biology that I just want to remind you of it and also to show you the notation that I'm using. So key concepts that we're going to be using are variance, covariance, and regression. And you should be familiar with those concepts. The notation that I will be using, we're going to say the variance of some variable x, I will write it like this, double bracket x, close double bracket. The reason we're going to do this is sort of twofold. One is we're going to have combinations of things. If we go down the line, we have situations where there's a covariance between the variance of one thing and a regression of something else and something else, right? And if we write out these terms, it just gets impossibly long and hard to interpret. Also, we're going to be using different kinds of statistics-like operations. So this particular will be what I'll call the frequency variance, and that is the variance over a set of things in a population right now. Okay, and I'll make that clear as I go along what that means. In other words, not over a probability distribution of possible future outcomes. We're going to write that differently. Covariance, we'll write like this. The covariance of x with y, we'll write as double bracket x, y. Okay, and regression. And when we talk about regressions here, essentially we're talking about a least squares linear regression. I'll write them by stacking them. Double bracket x on y. That'll be the regression of x on y. That's the way it's written in statistics when people say it. Okay. Now, I assume you're all familiar with these concepts. I just want to point out a couple of things about them. When we talk about the variance of something, a way to think of it is it's the mean of the thing minus its own mean squared, right? So we could write this as variance of x is x minus x bar squared bar. You're probably familiar with that nomenclature. When I put a straight line over something, that means a mean, specifically a frequency mean. When we do probability means, that'll be a different symbol. So this is one reason I'm being real careful with nomenclature here. One thing also about this, if you look at this from an intro statistics sort of perspective, we would write this as 1 over n, where n is the number of things, like population size in our case, summation I equal 1 to n of xi minus x bar squared. Note something, and I don't know if this will stand out to anybody here. To some of the biologists, you might look at that and say, why isn't there an n minus 1 down there? Okay, because, and this is something you have to watch out for. In statistics, we're not doing statistics here. We're talking about variances and covariances, but they are just mathematical operations on distributions. We're not doing statistics. Statistics is about working with samples and inferring things from samples. And statisticians often, when they talk about the variance of something, they mean the variance of a large population of things that you're trying to estimate from a small sample that you took. And in that case, there's a bias in the variance of your sample versus the variance in the whole thing. And one way to approximately minimize that bias, it does not eliminate it, but it minimizes it, is to divide by n minus 1 here, okay? But that's not what we're doing. We're not drawing a sample and then trying to infer a property of a whole from the sample. We're just calculating the actual variance in a population. Okay, so do that. One reason I emphasize that is not only in intro statistics classes, it's often just presented as though you divide by n minus 1. And if you look at a lot of software, like Microsoft Excel, I think does this. They have built-in functions to take like the variance of a bunch of things. You can give it a bunch of elements in a column and it'll take the variance of them. It does it as a sampling variance. So it divides by n minus 1. I always tell students when we're not doing homework here, but when I'm doing a class with homework, do it on paper. Don't use Excel to do it. And I can tell the ones who did because they get the wrong answer, because Excel treats it as though you're doing statistics, not just pure mathematics with a distribution. So that's just something to keep in mind. You should have an intuitive sense of what variance is about. It is one way of measuring the spread of a distribution, right? So if I have a distribution like that and another one like that, the broad one has a higher variance than the narrower one. So just when you see it and it pops into your mind, you should be thinking of that. Similarly, covariance, this here, covariance x and y is basically x minus x bar times y minus y bar, average over it. And if you were calculating this given a bunch of numbers, again, you would divide by n, not n minus 1 in that case, right? And regression, this is another one where if you've had a statistics class, you might get a skewed, pun not intended, idea of what regression is. When we're talking about a regression here, it is the slope of a least squares linear regression. So if I have, say, I did it of x on y, so I'll do it this way, x, y. And if I have a bunch of values like that, if you pick the least squares best fit curve through that, the slope of that is the regression of x on y. Okay. It is the slope of the best fit straight line. As we're going to see, though, that does not really limit us in the ways that you might expect it to. Okay. Key thing about these. So just some basic properties of these that you probably remember, but bear repeating because we're going to be using it a lot. The regression, I'll write it in the same order there, of x on y is equal to the covariance of x with y divided by the variance in y. Okay. So when you regress on something, you're regressing on the thing that you're putting the variance down there. Variance, of course, is the same as the covariance of something with itself. So this is a covariance of x and y divided by the covariance of y with itself. That might look familiar to the mathematicians in the room, and it should especially look familiar when you think about what we're doing with this. I'm putting a bar over it, but you can think of that as basically I'm integrating over it. It might be discrete or it might be continuous. By the way, everything we're doing here applies to discrete and continuous functions and distributions equally. I'm essentially taking an inner product, okay? A covariance is an inner product. And so we're taking the inner product of x with y over the inner product of y with itself. What's that? That's a projection, right? If you're projecting x onto y, you write it as the inner product of the two divided by the inner product of one with itself, okay? So, and this is, you don't need to know this or think of it this way to understand the biology that comes out of it, but for the mathematicians, when you think of a regression, think of a projection. That's what we're doing, is projecting something like that. Okay, so that's just a bit of nomenclature, and mainly this nomenclature, this double bracket symbols that I'm going to use throughout. So you will get used to seeing that as we go through. Now, what we want to do is say, if we make no simplifying assumptions whatsoever about a population, can we draw some conclusions, some mathematical conclusions about rules that the evolution of that population has to follow, okay? And we can. And to illustrate the basic idea, I'm going to write it like this. Let's imagine we have a generation, and at first I'm going to use the term parents and offspring, but really parents simply means ancestors. And I've got a bunch of individuals. I'm going to draw them in circles because it's convenient. Something like that. And each of these has a phenotypic value. And I will throughout use Greek phi as phenotype. What is a phenotype? It's any property of an organism to which we can assign a number. Everything we work with will be real numbers, but you can do it with complex numbers just as well, okay? And in fact, you can extend it to vectors and matrices and tensors, where they become arrays of complex numbers that you assign to a value of an organism. But for now, we're just going to be talking about real valued traits. So it's basically anything you can assign a value to. So that includes what's traditionally called phenotype, height, weight, something like that, how altruistic you are to other individuals, those are phenotypes. At a locus, whether something is allele A1 or A2, that's a phenotype. Similarly, any function of any of those things is a phenotype. So an organism's body mass cubed raised to the power of its basal metabolic rate and divided by whether it has an A1 allele at a certain site, that's a phenotype, right? So any function of those also is a phenotype. So it's a very broad concept. And so this individual, I'll say it has phenotype 1, phenotype 2, phenotype 3, phenotype 4, like that, okay? That make sense? Yeah, question?
You said sometimes, well, you led me to think that sometimes one describes phenotypes with complex numbers.
Yes, you can.
Which kind of situation would that be useful in?
The only case where I've ever made use of it is in talking about the developmental maps, like on a limb, because it's traditionally represented as this clock face thing. It goes around a circle. And so a natural way to do that, you can do it X and Y, but you can also treat it as the unit circle in the complex plane, and often do that. We're not going to do any complex numbers for this, though. And it really doesn't change anything when you do that. The math that you get, the equations are exactly identical. And incidentally, since you asked, we're not going to be using this. But since it came up, you might say, how do you talk about the variance and covariance of a complex number? The way you do it is treat it as a multivariate case, right? So real, imaginary, and you treat those as two different values. And then you are, instead of a variance being a number, it's a vector, right? A vector of the variance of real part, variance of complex part, covariance between them are the off diagonals. So it's easy to do that, but we won't have a reason to do it. Okay. So phenotype's a very generally defined thing. So we're just saying we've got a population of things with phenotypes, with traits. And they produce a population of descendants. And sometimes I'll call them offspring. But really what I mean is descendants. And that's a very general term. And it might look like this. This might produce 3 descendants. This might produce 2. This might produce 1. This might produce none. Okay? And each of these descendants has a phenotypic value. And so I'll call this phi. I'll use a superscript O, again, for offspring, but really it's descendants. That doesn't mean raised to a power. Superscript indicates that it is of the next generation of 1, 1, phi offspring 1, 2, phi offspring 1, 3. First index being the parent, second being the ordered individual here. This would be phi offspring 3, 1, phi offspring 3, 2, and this would be phi offspring 4, 1. And you notice I picked an ordering on this. This is purely for the sake of making painfully obvious how we're doing this derivation. The ordering is arbitrary, right? I could order these organisms any way I want. I could order these in principle any way I want. There are circumstances where you might want to say, wait a minute, I want to know the order of offspring. It matters if it's the first or second or third offspring produced by an individual. Biologically, sometimes that matters. In that case, the order of that offspring among the offspring of that parent becomes one of its phenotypes. Right? So phenotype can be something you measure. It can also be that you are the second offspring produced by those parents. That's a phenotype. And right now we're doing this with a single phenotypic trait, say something like body mass or anything you want, but we will extend this to multiple traits. And you'll see how that goes. So does this setup make sense? I just sort of described ancestors and descendants like this. You will note something. Each of these descendants comes from one and only one ancestor. That's not what we see in organisms that are obligately outcrossing, sexually reproducing. In that case, each offspring comes from 2 ancestors. In fact, we'll get exactly the same result down the line. The only thing is when offspring at a later stage, and I'll point out when this comes up, when offspring are a result of sexual reproduction between 2 separate individuals, then we count each offspring as 1/2 for a descendant for that individual. And the reason is purely accounting. It has nothing to do with how much genetic material is being passed on. That'll come in separately. But it has to do with the accounting, right? So if in fact this offspring were a descendant of that parent and that parent, when we calculate the fitness of these parents, we count it as 1/2 for that and 1/2 for that. That's the only modification we have to do for sexual reproduction. Okay, so this is a very general sort of scenario. What can we derive from it? Well, let's just write out the terminology we'll be using, and I've given you a bunch of it already. So capital N is population size. Population size of parents. And really, that means just ancestors. When I just do a phenotype by itself, phi is phenotype of parents. Phi with the superscript O is phenotype of offspring. In the most general sense, phi by itself is the phenotype of the population of things that you're starting with. And phi O is the phenotypes of things later, at some later time. By the way, and I'll mention this again as we go on, you can count an individual as one of their own offspring, right? So you might look at this and you might say, wait a minute, this is non-overlapping generations, right? These individuals gave rise to these and disappeared. Nope, mathematically, this could be that individual. They just survived that long, right? So your descendants include your offspring. It includes you if you survive longer. If you are an organism that's capable of asexual reproduction by cloning, it includes all the clones that you produce. Basically, all the descendants of you at some time in the future. And it could be any time in the future, but things like fitness will be defined relative to the time we look at. So traditionally, we look over one generation for the organism. Sometimes it's useful to look over more than one generation. Okay. We will not be working with cases like that, but you probably know if you're a biologist, you know probably of cases where you look over 2 generations. Anyone know a case where you count grandchildren instead of offspring in determining fitness? It's a problem in biology, in evolutionary biology, that we solve by counting grand offspring. Sex ratio. Fisher's sex ratio theorem, explaining why most populations tend to go close to a 1-to-1 sex ratio. Even if the breeding sex ratio isn't 1-to-1, most populations are very close to a 1-to-1 sex ratio. Why? Fisher's sex ratio theorem addresses that, but it does it by counting the number of grand offspring you produce, right? Because that'll be different depending on the number of the proportion of females or males you produce in your own offspring. So sometimes descendants can be more than one generation down the line. We won't be looking at that case. If anybody's interested in that and you haven't heard of it, I'll happily tell you about it later on or after class. But the point is, this notion of descendants is very general here. And then I'm going to specify, I already gave you the subscripts that we used here. I will specify phi offspring ij, I'll write it that way, is the phenotype of the jth offspring of parent I. And then, in fact, we're going to have reason to average over those, to say what's the average phenotype of the offspring of an individual. And so here we'll use just a notation that is borrowed from statistics sometimes. If I write offspring with a single subscript, okay, not 2 subscripts, a single subscript, that means I'm averaging over the second subscript. Sometimes in statistics you just do this. Some statisticians put a dot there to mean that you're averaging over that. It's just a notational convention. So this is the average phenotype of individual I's offspring. And all this will come up in doing our derivation, which once I write these nomenclature up will go very quickly. And then for the most part, we won't be writing subscripts on those. Those won't be necessary. And finally, I'm going to specify another symbol, lowercase delta. And lowercase delta for I is the difference between their offspring's phenotype and their phenotype. So that's phi offspring I minus phi I. And this gives you no more information than specifying both of those. But we'll see sometimes it's useful to think of how different is the offspring from the parent. And other times it's best to just think of what's the phenotype of the offspring. So I'll put that in to do it. And then the other term that I'll use that we haven't discussed so far, we're going to be talking about selection now. That's one of the things that can be happening. And so we have to specify fitness. W is fitness. And if I say W subscript I is fitness of individual I. So it is a property of an individual organism. How do we measure fitness? It's the number of descendants that you produce. That's it for this discussion. Number of descendants. And for our purposes now, we'll be talking about over a single generation. But for organisms that can potentially live a very long time, like humans, like a number of things where you have overlapping generations, this means you count the probability that you survive that long as part of your own fitness, right? So even if, and this makes sense, you've got a big forest adjacent to here, just as you go inland from here, you've got a huge forest with a lot of trees that live for a very long time. And many of them, they're always putting out pollen and ovules, and they're always potentially doing reproduction, but many of them actually do very little reproduction in most years. It's a few certain years they do most of their reproduction. So they may survive for decades and produce practically no descendants, but they still get to count their survival for that amount of time as their fitness, as contributing to their fitness over that time. Okay? Humans, among animals, are pretty similar. We live a very long time, much longer than the average time to first reproduction. Not as extreme as some of the trees, but compared to most animals, we do that. So fitness is defined very broadly in this case. Okay, so with those things in mind, what can we conclude? Well, what we want to do is figure out what is the mean phenotype. Ultimately, we'll want to know what's the change in mean phenotype over this time, okay? And to get at that, I'm going to ask, what is phi bar prime? And that's mean phenotype after one generation. Okay, and I'm not using a superscript O there because this is just taking the mean over all, but it is taking the mean over all these individuals. But it's sort of like after all this reproduction has happened, survival and reproduction has happened, there's a new population. What's the mean phenotype of that population? I'll write that as phi bar prime. Okay? So, with that in mind, how do we figure it out? Well, it's not really hard. I'm going to leave that up. Everybody have this down? If I pull this over, then that's obscured, and I kind of want to refer back to that picture. Everybody okay if I erase this? Okay. Y'all have the advantage of cell phones, which is great. I recall as a student so many times just frantically writing. It's like, he's erasing it! And I have to keep writing. And sometimes I would make an agreement with my roommate. Like if it was a class that had a lot of complicated diagrams, one of us would draw the diagrams and the other would be taking notes, and then we'd put them together because you could not draw the diagram and take notes fast enough. That's one reason, by the way, I always lecture with a board. I do not use PowerPoint, okay? Or the old-fashioned version of PowerPoint was overhead slides. So you'd have these transparencies and you put them up and something. But you could put up a transparency with the life cycle of some sort of bryophyte, right? This really complicated life cycle. And you can say, look, this, pull it off before the students could write it. If I write it on the board, you have time to write it in your notes. So what are we after? We want to get phi bar prime, mean phenotype in the next generation. And I'm going to sort of draw this out. It'll go quickly, but I'm doing steps that once you see it, you think, oh, I could skip steps. I'm not going to skip them, though, because I want to drive home just how sort of fundamental this is, and the fact that we're not introducing any simplifying assumptions. So I can write this as what? It's the sum of the phenotypes of all these individuals divided by the number of them, right? So I'll write it as this, summation I equal 1 to n, summing over all parents. And then within that, summation j equal 1 to w sub I. So now I'm summing over these. So the first one, I do parent 1, and then I do summation over 1, 2, 3 of its offspring. And then I go to parent 2, and I sum over its offspring, it has none. Go to parent 3, sum over its offspring. So all I'm doing is sequentially going over each of these, right? So that should be straightforward. It's that of phi offspring ij. That is the sum of all those phenotypes. And of course, we have to divide that by the total number of them, right? Well, what's the total number of them? Just the summation I equal 1 to n of w sub I. W is the number. This one's w is 3, this one is 0, this one is 2, this one is 1. Okay? So all I'm doing is just essentially adding up all these numbers and dividing by the number of them. I'm not assuming anything, I'm not doing anything complicated with it. I want to note something. A couple of things that when you see them, it's obvious after the fact, but when you see them, you can really simplify this. One thing is, what's mean population fitness, W bar? Well, that's just 1 over n, summation I equal 1 to n, wi, right? So, summation I equal 1 to n of wi is just n w bar, right? And I've got that summation down here, so that's kind of convenient. And similarly, what's the mean phenotype of I's offspring, right? So that came up when we talked, when I put up those symbols, the mean phenotype of I's offspring. That's just 1 over w sub I, summation I equal 1 to w sub I of phi offspring ij. Obvious. So summation yeah, 1 over wi. This should be j, pardon me. J equal 1. Thank you for pointing that out. So this is just tells me the summation of j equal 1 to wi of phi offspring ij is just phi offspring I wi. Question?
Oh, sorry, I don't get what is the phi, like it's like sequences when you're trying to sum over one phi, because you're talking about the phenotypes? Yes.
These phis, yes. So this is phenotype. If there's a superscript O, that means it's in this generation.
Yes, but right. What is this phenotype? Like, I mean, sequence of nucleotides, it's a number,
it's anything I can assign a number to.
Okay.
So, body mass. This could be the body mass of all these and then the body mass of all their descendants.
Okay.
It could also be, for an allele, it could be whether or not you have an A1 allele.
Okay.
Okay, if this organisms are haploid, then each of these is a 0 or a 1. A 1 if they have A1, a 0 otherwise. Each of these is a 0 or a 1, right?
Okay.
If they're diploid, we're going to do this later, we do it as the frequency of the A1 allele within their genotype, in which case each one of these is 0, 1/2, or 1.
Yes. Okay.
And then each one of these is 0, 1/2, or 1.
Okay.
Okay. If it is a nucleotide, so this is a an added complexity that we're not otherwise going to focus on here. If you're talking about nucleotides, so it's an A, a C, a G, or a T, in that case you represent it as a vector, okay, rather than as a number. And I said we can apply all this to vectors and other sorts of tensors. This is just, you don't even have to write this down because we're not going to be doing this case. But if you're using nucleotides, those are essentially categorical variables, in which case an A we write as a vector 1, 0, 0, 0. A C is a vector 0, 1, 0, 0, and so forth. Okay. We're not going to be using that case. So all the cases we look at will be things you can assign a number to. Any other questions on this? So by doing this, oh yeah.
Well, I didn't understand. I actually have a mic. Could you explain like what's the logic between the phenotype after one generation equation? Because I didn't get why you divided by the fitness of the parents and not like its own fitness or the number of offspring.
Okay, so fitness of parents is number of offspring.
Oh, okay.
So for this individual, W1 is 3, W2 is 0,
Okay.
W3 is 2, and W4 is 1.
Okay. Okay. Now I get
Make
it.
sense?
Thank
Sorry, I
you.
should have written that up there. That would make it clearer. That make sense? So yeah, we're just adding everybody up and dividing by the number. Okay. Like I say, this is so painfully obvious that you might be thinking, why is he even bothering to do it? But as I'll emphasize later, that's actually part of the point, is how painfully obvious it is. So having done this, here we've got this. This summation is my denominator here. This summation is that summation in there. So I can write this as 1 over n w bar, because that is just n w bar, right? That denominator. Summation I equal 1 to n of wi phi offspring I. Right? And what's this? Summation I've got a 1 over n, ignore that 1 over w bar for the moment. I've got a 1 over n, summation I equal 1 to n of a thing. What's that? That's just the average. That's the mean over n, right? So this is just equal to 1 over w bar times phi offspring w bar. The average of the product of the average mean of your offspring times your fitness, averaged over all the individuals in the population. And when I talked about variances and covariances and things earlier, I didn't remind you of this, but important things to remind you of. You probably remember this from intro statistics, but again, this is something we're going to use a lot. If I have a variance, or a covariance, I'll say, of A and B, that's equal to the mean of AB minus the mean of A times the mean of B. That relationship is going to come up all over the place. And doing the calculations that we're going to be doing, this is absolutely fundamental to remember. Okay? If you're not familiar with it, if you've not seen that before, ask me after class and I can show you how to derive it. It's easy to derive. Or if you're thinking, I don't remember that, but it makes sense and you want to go home, I'll give you hints and you want to go home and try to do it on for yourself, that's fine. If you remember it, good. Definitely, though, be aware of this. We're going to use it a lot. Something about this. We have a special notation for things like variance and covariance because those terms come up everywhere in evolutionary theory. Once you start talking about selection, is it okay if I close this? As soon as we start talking about selection, covariances are ubiquitous. Basically, try understanding basic mechanics without knowing what a time derivative is, the derivative of x with respect to time. It's practically impossible. Try and understanding basic, just Newtonian mechanics without the basic idea of a derivative. Okay, that's like trying to understand evolution without covariance. It is that central to evolutionary theory. So we have this relationship, and this of course means that AB bar is equal to the covariance of A with B plus A bar, B bar. And that's what we're going to be using here, because I have here offspring phenotype times fitness bar, right? So this is equal to 1 over W bar times the covariance of offspring phenotype with fitness plus offspring phenotype bar fitness bar, right? And note I've got a 1 over W bar here and W bar here, right? So that's really, I can write it as 1 over W bar, covariance, offspring phenotype with fitness, plus mean offspring phenotype, because that W bar cancels out with that one. Make sense? And now what we really want is the change in mean phenotype. Change in mean phenotype over this time. And I'll write that capital delta phi. And what is that? That's just phi bar in the next, among the offspring, minus phi bar. Right? So using this, I could write it as delta phi, this should be delta phi bar, is equal to 1 over W bar, covariance, offspring phenotype with parent fitness, plus, I'll write it out, plus phi offspring bar minus phi bar. And here, remember we use that lowercase delta symbol? That's, the average of that is just this. So ultimately, I can write delta phi bar is 1 over W bar, covariance phenotype of offspring with fitness of parents, plus that lowercase delta bar. That's our result. This was originally derived in a slightly different form, but this basically result was derived by George Price in 1970. This is called the Price equation, or Price's theorem. I'm going to emphasize the term Price's theorem because this is a basic mathematical property of any evolving system. We made no simplifying assumptions in deriving this. I did not assume population size is fixed. I did not assume there's random mating. I did not assume any value is small enough that we can ignore the squared terms, right? No simplifying assumptions. The only thing that biologically is an added thing that this did not include is migration. I assumed that all the descendants are descended from those ancestors. If you have a partially open population where individuals are migrating in and some are migrating out, that is something we would have to add separately. I can show you later how we add that in. You get a general result similar to this. Basically, you end up with, instead of 2 terms, 4 terms. 2 of them have to do with what's going on within, and the other 2 have to do with migrants coming in and immigrants going out. Okay? But for a closed population, if you think of it as for a species, right, a reproductively isolated unit, we made no simplifying assumptions here. So this is true. And what I mean by saying it's true is basically a law of nature. All evolving populations anywhere in the universe have to obey this rule. And something to note about it, you look at covariance. One issue that some people had with this when it was first published, and even a while after that it was first published, was, well, covariance, that's really kind of like measuring a linear relationship between things. What about nonlinear relationships, okay? They don't come in here. I want to point out, remember you can write a covariance, you can write a regression in terms of a covariance and a variance. You can write a covariance in terms of a regression and a variance. So I could also write this as delta phi bar is equal to 1 over W bar times the regression of fitness on offspring phenotype times the variance in offspring phenotype, right? I'm just breaking up that covariance, and then I'd have to add delta bar. Yes?
I'm just curious because there it's initially W times the phenotype, then in the mean you have the phenotype times the W, and then it's the covariance of the phi, then the W. If we did it like in the other.
Covariance is symmetrical.
Okay.
Covariance of A with B is the same as covariance of B with A.
Okay. Okay.
That's central to this.
Okay.
And you can tell from this because multiplication is commutative for what we're dealing with here, okay? There are cases where kinds of multiplication are non-commutative, but we're dealing with multiplication being commutative, so I could reverse these and I could reverse these. So covariance is symmetrical. I could do A with B is the same as B with A. Regressions are not symmetrical, right? Because there you're dividing by one of the variances, but not the other one. So that's important. For regression, the order totally matters. Okay? Now, it should make sense from the definitions I use. This is equivalent to this. And this brings up an issue that is kind of surprising to people. Let's say, for the moment, that all the deltas equal 0. This is just to illustrate a case. Then I have delta phi bar is 1 over W bar, regression, fitness on phenotype, because I set the deltas equal to 0, times the variance in phenotype. Remember, that regression, that's a least squares linear regression. So when you visualize it, you tend to think of something like this. This is a regression of fitness on phenotype, so I put phenotype down here, fitness up here. Say I've got a bunch of values that look like this. Then I've got a regression line that should be a straight line. That's just my bad drawing. Going through it, and the slope of that regression, that combined with the variance in phenotype is what's determining the change in mean phenotype. Okay, but, and here's where we're doing something different from statistics. If you look in a statistics book, they'll say, be aware. Least squares linear regression doesn't always capture what you want to capture, right? Because I could write a different, very different looking distribution. Let's say something like this. For putting a bunch of values. Something like that, this J-shaped distribution. I could shape that so that it would have exactly the same least squares linear regression, right? And statisticians use that, I meant those to be the same slope. Statisticians use that to say, see, least squares linear regression doesn't capture everything you want. But think about it, we just derived this, making no simplifying assumptions whatsoever. In fact, if the joint relationship between phenotype and fitness looks like this, and if the variance in phenotype is the same in these cases, which I could add some points to make it, then in fact, in these two cases, the change in mean phenotype would be identical. Okay, and that's a key here, and that's a key to understanding how this is different from statistics, because we're treating this regression just as a mathematical property of a set of points. The way you can do a regression on a continuous function just perfectly well too. So I could have drawn a line, and then I would have the best fit straight line through it. So what we've concluded, because we put no simplifying assumptions into this, is that if the regression slope and the variance of phi were the same in these two cases, then in fact the change in mean phenotype would be the same. And you can check that, right? You can make up cases. I remember doing this, sitting down, making up, okay, I'm gonna make up a really weird case. But, so I've got points like in some weird direction doing some strange function here, but so that it has the same variance and the same slope, sure enough, the change in mean phenotype is identical. Okay, so when I say this is a law of nature, like other laws of nature, it's very useful because it tells you what things you have to look at, and it also tells you what things you can ignore, depending on the particular thing you're calculating. Okay, does that make sense? So if these slopes are the same and if these variances were the same, then the change in mean phenotype would be the same in these two cases. Now here, and this is the thing that is a difference between building a theory like this and doing model building. Because in model building, you start out with simplifying assumptions and then you go on. And if you get some weird result, like, wait, that can't be true, that doesn't seem right, immediately the thing that comes into your mind is, okay, one of my simplifying assumptions wasn't appropriate, right? There was something wrong with my assumption that this value is so small I can ignore squared terms. Or maybe there was something wrong with my assumption that population size is constant, or something like that. So if you get a weird result, you sort of don't know how to interpret it. It might just be a result of your assumptions. In this case, we made no assumptions. And so when we get a surprising result, it's true, okay? Now, I say we made no assumptions. We made no simplifying assumptions. We did make some assumptions that are essentially definitions. I talked about ancestors and descendants. And I said that they have phenotypic traits that you can measure with a number, right? And I said it could even be a complex number for our purposes. Let's just say it's a real number. You can measure the phenotypic trait with a number. I also assumed, I'm summing over things here. It wasn't obvious, but in some of these sums, I assumed the population size is finite, right? That it's not an infinite series. Okay, so those are simplifying assumptions, right? We can identify ancestors and descendants. We can assign phenotypic values to them. We can define phenotypes such that we can identify the same phenotype in the ancestor and the descendant. That's an assumption. Populations are finite, right? They could all be captured with a finite number. It could be an arbitrarily large one, but hey, they're finite. It's not a trans-finite number. There are no alepphs in here. Those are assumptions, but they're assumptions that we have reason to think are exactly true of all biological systems. Okay, when I say, I'm going to assume all populations are finite, most people would say, I buy that that's actually true of all populations in the universe, right? Anywhere you go in the universe, because what's the estimate? 10 to the 82 or something particles in the universe physicists come up with. They come up with estimates for the number of particles in the universe. It's a very big number, but it's a finite number, okay? The universe is finite, and so populations of things in it have to be finite. So the assumptions we're making here are really things that we think are exactly true. As such, they are more appropriately, I think, called axioms rather than simplifying assumptions. And that's a term, axioms. It gets used very sloppily by scientists sometimes. Sometimes scientists say, oh, this is axiomatic, but what they really mean is everybody sort of agrees that it's a good approximation, okay? I'm not saying that. I'm saying things that we honestly think are exactly true. And so in a sense, we're using scientific axioms to derive this. That means a weird result like this, that the shape of this distribution, I can really change the shape of this as long as the slope is the same and the variance in phi is the same, I get the same change in mean phenotype. That is true necessarily unless one of my axioms was wrong, okay? Now, in model building, if you get a result that, well, that doesn't quite make sense, one of my assumptions was wrong, well, you know your assumptions were wrong. You know population size isn't constant over time. You know that these organisms aren't doing exactly random mating. You know that mutation happens, things like that. In a physics context, you know that friction happens, okay? You were just assuming it didn't for this calculation. So if you get a weird result, you assume, okay, there was an error in one of my simplifying assumptions. Get an error here. If you check it back and you make sure, yeah, it works, the change in mean phenotype should be the same in these cases. And then you worked out a case and it was different. You'd say, wow, one of my axioms is wrong. And if one of your scientific axioms is wrong, that's big news, okay? So one result of building theories like this is when your scientific axioms, if you did get a result that actually was wrong, that would tell you something big. Whereas in model building, you get a result that's not exactly right, you never expected it to be exactly right, you know? So there's a different way you think about these things, a different way we think about how to interpret when our theories work and when they don't work. Now, just a couple of things to emphasize the generality that goes with this. I've emphasized already, but it bears repeating, phenotype can be anything we assign a number to. If, you don't have to write this down, but because it came up as a question about the symmetry there, and I said, well, of course, multiplication is commutative, right? Is multiplication of tensors commutative? Generally not, right? If I multiply two matrices together, the order matters, okay? And remember, I said when we're talking about nucleotides in a DNA sequence, then we're representing them with vectors. And if you want to talk about a pair of nucleotides or three nucleotides, they are higher and higher ranked tensors in that case. In those cases, order matters. All the math that we do comes out the same. You end up with equations that look the same, except you have to be very careful about the order in those cases. But for scalar values, right, values that you can describe with a number, then everything here is commutative. So phenotype is very general, and also ancestor-descendant is very general. And I already mentioned this, but it bears repeating. When you talk about the phenotypes of ancestors and descendants, you can count yourself as one of your descendants at a particular time in the future, okay? And the only thing I'll add is, and this comes into the accounting, when you have some individuals reproducing sexually and some individuals reproducing asexually, then when you're calculating fitnesses, you count each sexually produced offspring as 1/2 contributing to their fitness. So when you're looking at the fitness of this individual and they have 2 sexually produced offspring, their fitness is 1 because you count each of those offspring as 1/2. Okay, that's the only modification here. You don't have to do that if you're dealing with things that are always reproducing sexually. Why not? Because we could do it. We could put a 1/2 in front of each one of these, and then we put a 1/2 in front of each one of these, and the 2's cancel out. So if everyone's reproducing sexually or everyone's reproducing asexually, then we just count offspring as offspring. if some individuals are reproducing sexually and some asexually, then is the only time that you have to be careful and count sexually produced offspring. And I mean sexually by outcrossing. Not selfing, you count them as one. But outcrossed sexually reproduced offspring, then you count them as 1/2. Okay? But again, that only matters when different kinds of reproduction are happening. Because if everyone's doing that, you get a factor of 1/2 on the numerator and denominator, and it cancels out. Yeah?
How does this look like when we're counting overlapping generations, when we're counting the parent as one of the offspring?
Yeah, then you would count the parent as 1, and then you would want to count sexually produced offspring as 1/2. Any other questions? Yeah.
I don't know if I can formulate like a question very easily, but it's for me the interpretation of this phi prime bar and phi o bar. I know that they're different objects because one you're averaging the phi ij and then you have the over the entire offspring population, and the other you're averaging the average offspring phenotype over the parents, right? The number of parents.
Mm-hmm.
But especially this phi o bar, for me, there's not a clear interpretation for what this thing actually means, right?
Okay, so you mean the difference between that and that?
Yeah, in terms
Yeah.
of like an intuition. I don't
Yeah,
have
so this is averaged over all parents. So I have a mean offspring phenotype, right? I have 3 kids, so you'd average them to get my phi offspring. And somebody else might have 4 kids. They'd average them and get their mean. In calculating this, I'd add mine to theirs and divide by 2. So the fact that they had 1 more kid than I do wouldn't come into it. Whereas in this, we're taking the mean over the entire descendant generation. So we'd be counting my 3 and their 4. And then that would go into this. So it would be a different value. Does that make sense? Yeah. Any other questions on that? Okay, so how do we apply something like this? One of the objections that some people had to thinking of evolution in terms of something like this is, okay, that's interesting, it's this very general statement, but how do we actually apply it to a particular case? Well, if we have a general law like this, when we apply it to a particular case, that's when we bring in, in a sense, simplifying assumptions, because we say, okay, I'm talking about this kind of phenotype, and that constrains what we do, right? So you still bring in assumptions to apply to a particular biological system, but they come in after we've derived the general result. Also, I didn't mention, but I want to be clear on, how do you interpret these 2 parts? Note, this first part has fitness in it, okay? So this term captures all processes that involve differential survival or reproduction. All processes involving differential survival or reproduction. Okay, so that includes selection. That also includes drift. Okay, we're not going to be spending much time on doing drift from this. We focused on drift using a particular model, but you can derive drift equations from this. It's more complicated than doing it using coalescent theory, because coalescence puts in all these nice assumptions that makes it easy to do, but you can do it, and drift comes out in this term, okay? Because drift involves differential reproduction or differential survival. It involves random variation in how many descendants you produce, okay? And so it means some individuals might produce more offspring than others. If that happens randomly, that contributes to drift, not selection. But it's still differential survival or reproduction. This term involves changes that occur in the process of reproduction. Changes that happen in the process of reproduction. Examples that we'll be looking at for there are mutations. Okay? Mutation, in general, doesn't have anything to do with how many offspring you produce. It's something that just happens when you're producing gametes, usually is occurring at meiosis. When you're producing gametes for the next generation, some mutations occur. So mutation will go in here. So this includes things like selection and drift. And this includes things like mutation and recombination. So that gives you some idea sort of biologically where pieces go in there. Okay. Other things that could go in this term, anything that uniformly just affects the whole population. So a uniform environmental change, for instance. For a lot of marine organisms, for a lot of marine invertebrates, if you raise the water temperature, they develop faster. That's just a consequence of developing in a higher temperature, right? So if the ocean temperature comes up, the entire population will start developing faster. That would go into this term, right? It has nothing to do with differential survival or reproduction of individuals. It just means this thing that's affecting everybody is going on. So environmental changes that affect the whole population as a unit are in here, but they go into this term here. Okay. So with that in mind, how do we apply this? And we could apply it to population genetics, we could apply it to sequence evolution, we could apply it to phenotypic evolution, what's traditionally called phenotypic evolution, and things like change in quantitative traits, like body mass and basal metabolic rate and things like that. We're going to start out using population genetics as our example. So we looked, when we were talking about coalescent theory, we looked at the effects of drift on things like allele frequencies, right? We calculated things like the probability of fixation of an allele, right? Coalescent theory is great for that, as long as there's no selection acting, okay? Because a key property, a key assumption of coalescence was that IID assumption, that everyone's distribution of descendants, every gene copy's distribution of descendants is independent, identically distributed. That's obviously not true when selection's happening, right? When selection's happening, some individuals are producing more descendants than others are. Is this locked? I'll just close it part way. Folks have gone out. So we cannot use coalescent theory to address selection. Incidentally, we made a bunch of other assumptions in coalescence. For instance, also we assumed there was no immigration or emigration. Somebody asked a question the other day about recombination. We were assuming there was no recombination within loci. People have extended coalescent theory to include all those things. They are still trying to figure out how to extend coalescent theory to include selection. It just doesn't work, right? You lose all the simplicity that makes it appealing when you put selection in coalescent theory. So we can't really apply it to that. So we're now going to talk about changes in allele frequencies that involve selection. And we're going to derive our results from this equation. Okay, so we're going to consider a case of one locus with two alleles. If you have one locus and two alleles, there are 3 genotypes. Those are, I'll call them A1, A2, and A2. Okay, so the one locus will be the A locus, and the 2 alleles will be designated A1 and A2. Yes?
Could you just repeat why we can introduce selection into the Coalescent Theory?
It turns out that assumption that the number of descendants was independent, identically distributed for each gene copy, you can't relax that without losing all the things that allow you to do coalescence, make it simple. Here's one of the sort of an intuitive idea for that. Remember I drew things like, I said, here's some individuals, and they each have 2 gene copies, right? And then in coalescence, I just ignored the individuals and said, okay, let's just look at the gene copies. I didn't have to worry about the fact that this gene copy is in the same individual as this one. When selection comes in, we do, because the thing that has a fitness is this individual versus this individual. So the basic trick of coalescence is to ignore individuals and just look at gene copies and their lineages. You can't do that when selection's acting. You have to pay attention to which gene copy is in an individual organism with which other ones. And like I say, you might be thinking, yeah, I'll bet I could still come up with a way around it, right? You're not alone. A lot of people have thought, I'm going to come up with a way around it, and to date, nobody has come up with a way around that problem. So I just let you know that. So does this make sense? We're talking about a locus, 2 alleles, 3 genotypes. We're going to assign fitness to these genotypes. Call this W11, W12, W22. Okay, so just assigning fitness values to it. For the moment, I am treating these as deterministic. In other words, I'm going to assign a number to it. We will modify that. Tomorrow sometime, we will change it and say each of these is a random variable and it has a probability distribution of possible values. Okay, and we'll see how that extends our results. But for now, I'm treating these as deterministic values. And when we do this, we need to assign, if we're going to apply Price's theorem, we need to assign phenotypes to them, right? Well, straightforward way to do that, those are the genotypes. Our alleles are just A1 and A2. And in general, I'm going to designate the frequency of A1 will be written lowercase p. That's just standard in population genetics. What phenotypes go with these? We have to assign phenotypic values. The way we'll do it, we're going to be looking at the frequency, change in frequency of the A1 allele. So I'm going to say phenotype of A1 is 1, and phenotype of A2 is 0. When we look at genotypes, we can say, what's the phenotype of them? Here, the phenotype is going to be the frequency of A1 within the genotype. So here, it's 1, because it's 2 A1s. Here, there's 1 A1 and 1 A2, so the phenotype will be 1/2. And here, there are no A1s, so the phenotype will be 0. We can show that these are identical definitions by saying really what we're doing is defining your phenotype as the frequency of A1 within the thing that we're looking at. If you're just looking at one allele, it's either 1 or 0. If we're looking at a genotype, it's 1, 1/2, or 0. Okay. Why do we do that? Because regardless of whether we're looking at alleles or genotypes, if we do this definition of phenotype, then the mean phenotype is P, the frequency of the A1 allele. And you can confirm for yourself how that goes. It's easy to see it in this case, right? Or if we just look at individual gene copies and say, is it A1 or A2? And we count 1 if it's A1 and 0 if it's A2, the average of that is the frequency of A1. Okay? Turns out the same thing goes here. If we count each of these as 1, each of these as 1/2, and each of these as 0, and we take the average of these, we get the frequency of A1. Why are we doing that? Because Price's theorem gives us a change in mean phenotype. But that means, since mean phenotype is P, we can use that to get the change in P, the change in allele frequency. Make sense? Now, like I say, when we apply this to specific cases, that's when our assumptions come in. So we derived our general result. Now I say, I want to talk about this case, so assumptions are coming in. So one assumption is I can define phenotypes this way, right? It is also conventional in basic one locus, two allele population genetics to make some other assumptions. So let's start out. We'll change this in a moment. We'll assume no mutation. Okay? And given that what we're really following is alleles from one generation to the next, if there's no mutation, that means that the descendants of that allele are the same allelic type as it was, right? That means, therefore, lowercase delta equals 0. Or for alleles, phi offspring equals phi. That'll change when we bring mutation back in, but it'll actually be easy. I'll show how it makes it easy. But if we do those, then this simplifies a lot. That term disappears, and this just becomes phenotype. So I get delta phi bar is 1 over W bar covariance phenotype with fitness, right? Because these are equivalent, and I don't have that delta bar over here because delta is 0. In other words, no mutation. And phi bar is P. So I can write delta P, change in allele frequency, is 1 over W bar. And now I'm going to write this as a regression. Regression, fitness on phenotype, times variance in phenotype. That's what Price's theorem turns into when we define phenotype in terms of what kind of allele you are, and we assume no mutation. Okay, then it just collapses to that. And note something. Regression of fitness on phenotype. What is that? That's the degree to which, as I change phenotype, fitness changes. Okay, that's selection. That's intuitively a very straightforward way of representing selection. So this regression here is capturing selection. And in fact, when we extend beyond one locus, two alleles, when we talk about quantitative genetics, everything will keep coming up with terms that have regression of fitness on a phenotype. And intuitively, when you see that, you go, okay, that's selection happening right there. It's the degree to which changing your phenotype changes fitness. Okay? So, how can we work with this? Turns out it's really easy to work with. It's going to be useful to focus on the allelic interpretation here. Keep losing my eraser. In the case of genotypes, I have assigned fitness to genotypes. Okay. You can assign a fitness to an allele. But to do it, what it really is the average fitness that that allele would have over all the genotypes that it appears in. And so I'm going to designate that W with a star here. That is not standard notation. And so this is W1 and W2. Standard notation in population genetics literature would just to be to use W1 and W2. And you say, well, how do you know you're talking about allelic fitness? You know it because there's only one subscript, right? If there are two subscripts, you say, oh, we're talking about genotypic fitness. If there's one subscript, you're talking about allelic fitness, okay? And you guys could pick up on that really fast, but I just sort of as a convention, I like to put a star there to indicate that this is the mean fitness of an allele across all the genotypes it appears in. Okay, but we could drop the star and you would know that having just one subscript tells you that fitness value. Okay, so how do we apply this equation? I'm going to erase this side. It's easy when we think about it in terms of allelic types because there are only two. We've got a regression of fitness on phenotype. How are we going to calculate that? It might seem it'll be complicated, but it's not. It's really easy when we're talking about alleles because there are only two, right? So what's our phenotype? Phenotype is 0 or 1, right? The phenotype of an allele is 0 or 1. Put stars on that. And what's fitness? Well, it's those marginal fitnesses. So this one might be W1, or no, this would be W0, W2 I should use. W2 star, because this is the fitness of the A2 allele, right? And its phenotype is 0. And this one might be up here. W1 star. Okay, and I could have put those anywhere. Now, what's the regression going to be? It's easy to find a regression when you have only 2 points. Okay, the regression is the slope of that line. That is the regression of fitness on phenotype. Okay, and what is that? Well, it's just this distance divided by this distance, but this distance is 1, 0 to 1. So that is simply equal to W1 star minus W2 star. The marginal fitness of allele A1 minus the marginal fitness of allele A2. We could do this, and you'll get the same result if you use genotypes. But then it's not visually as clear. You have to calculate the slope of that regression. Here, it's easy because it's just 2 points. So we're going to do it with that. So we have that. We also want to know the variance in phenotype. Okay? There are 2 types of alleles. And I'm drawing one copy, in a sense, at random, right? Because each allele is either A1 or A2. What's the variance? Do you remember? It's a Bernoulli distribution, or a binomial distribution if you draw only one thing. Remember the variance of that? I think I'm hearing P times 1 minus P. That's the variance for a single draw from a binomial distribution, and a binomial distribution, if you make a single draw, some people like to call that a Bernoulli distribution. So the variance is P times 1 minus P. Okay. So very quickly, we can write this out, and we get delta P is equal to everything's divided by W bar. The variance, P times 1 minus P, times this regression, which is W1 star minus W2 star. That is our standard equation in population genetics for one locus, two allele, change in allele frequency. You can find this in any textbook of population genetics. They will generally not derive it this way. They will generally derive it using these, and they will have to assume something about the frequency of these genotypes, so they'll assume Hardy-Weinberg frequencies, which assumes random mating, okay, which is an assumption that you can't always do. We got this from Price's theorem. The only assumption we added was no mutation. We didn't assume random mating. So this, in fact, is true whether or not you have random mating. Okay? As we'll see, if you have random mating or not, it affects how you would calculate these W1 and W2 stars. But in general, this is true regardless of whether you have random mating. Okay? Make sense? All right. I want to give you one more thing. We're almost done here for today. We'll carry on next time. But I want to give you one more thing that you can confirm for yourself. So like I said, in standard population genetics textbooks, they will assume Hardy-Weinberg frequencies for these genotypes. Now, by the way, anyone remember some of the assumptions behind Hardy-Weinberg?
I'm not sure if there's infinite size.
Effectively, they tend to, population size is big enough that you can ignore drift. That's one of them. Another one is no mutation that we did. Another one is random mating, okay? And another one is no selection, right? But we're studying selection. So how could you possibly use Hardy-Weinberg frequencies when you're imposing selection? Well, population geneticists are not stupid. They recognize that, okay? But they point out, with random mating, how long does it take to reach Hardy-Weinberg frequencies with non-overlapping generations and random mating? Anyone remember? 1 generation, right? So if you have 1 generation of random mating, you're in Hardy-Weinberg. So they use these, they focus on the zygote stage. They say, if you look at zygotes, what are the frequencies of genotypes in zygotes? And then the fitnesses are the probability that those zygotes survive to adulthood. And so even with selection, you can assume Hardy-Weinberg frequencies at the zygote stage. And so they'll assume frequency of zygotes. You know what I mean by that, right? Fertilized eggs. Then in that case, if we assume Hardy-Weinberg, this is p squared, this is 2p times 1 minus p, and this is 1 minus p squared. Right? So you got that. Now, here's what I'll give you guys to go home and think about. Sewell Wright, back in 1932, derived things differently. He didn't have Price's theorem, but he went through and he got a result that looked like this. Okay? And then from that, he derived another result for a special case of frequency-independent selection. Frequency-independent selection. What that means is that these fitnesses are not functions of P. That means Wij not a function of P. It could be a function of P. With frequency-dependent selection, your fitness depends on the frequency of strategies in the population. But let's say we assume frequency-independent selection. In that case, mean fitness, if we assume Hardy-Weinberg frequencies, is p squared W11 plus 2p times 1 minus p W12 plus 1 minus p squared W22. Where these W's right now I'm assuming those, that's actually true even with frequency dependence, but I'm going to now assume that those are not functions of P. If that's the case, Wright was interested in how does mean population fitness change as allele frequency changes, right? And so how do you get that? You take the derivative, dW bar dP. And when you do that, you can write it as 2 W1 star minus W2 star, where W1 star is the mean fitness of A1 across all the genotypes it finds itself in. So it's the probability that it's paired with another A1, which is P, times the fitness that it would have in that case, which is W11, plus the probability that it's paired with A2, which is 1 minus P, times the fitness that it would have them, which is W12. Similarly, we write W2 star is the probability that an A2 is paired with an A1, which is P, under random mating, times W12, the fitness that the heterozygote would have, plus the probability that it's paired with another A2, 1 minus P, times W22. Okay. If you take these definitions of marginal fitnesses, you can show that when you take the derivative of that with respect to P, it comes out to be 2 times this difference. Why does that matter? Because here's this difference right here. So check that. It's not an assignment. You don't have to hand it in. But just to solidify it in your mind, differentiate that and convince yourself that, in fact, it could be written in terms of these. And tomorrow, we'll start out with the result you get for frequency-independent selection. And I'll stop there for now. But take a look at that, and then it'll be ready in your mind when we talk about it tomorrow. We're done.